Okay, so let's move on to Masahito. Am I right?
Masahito.
Yamada. Welcome, Masahito. And let me introduce you a little bit. So you are one of the awardees. Masahito is one of the awardees, and he is sponsored by the Japan Agency for Medical Research and Development. And the title of his project, awarded project, is Study on Dietary Factors to Reduce Dementia Risk Related to APOE Genotype and Gender Difference. So let me hand on the floor to Masahito, and thank you for your presentation.
Yeah. Thank you, Jacqueline. Good evening, good afternoon, and good morning, everyone. I'm Masahito Yamada. I'm a neurologist serving as a director of Kudanzaka Hospital, Tokyo, Japan. Can I share my slides?
Oh,
okay. Okay.
Yeah, we have your slides on our master slide deck, so we can advance them.
May I have next slide? Yes, yes. This is my hospital in Tokyo. Yes, my clinical and research interests are in dementia and related disorders. I'm a 2020 Catalyst Awardee funded by Japan Agency for Medical Research and Development, AMED. Yes, the title of my project is a study on dietary factors to reduce dementia risk related to APOE genotype and gender difference, called DH. May I have next slide? Yes. As you know, the number of older people with dementia is rapidly growing. We aimed for dementia prevention with dietary factors. The work has started from our population-based cohort study for dementia called the Nakajima study. Nakajima is located here in Japan. It's a very beautiful town. In the Nakajima study, all residents of Nakajima aged 60 years or older are eligible for an evaluation of their brain function as well as physical health. Participation rate of the residents was 93% in the investigation of 2016 to 18. Dementia was found in 18% and mild cognitive impairment, MCI, in 20%. And among people with dementia, 67% had Alzheimer's disease, AD. May I have the next slide? Yes. It is well known that ApoE4 and female gender are major genetic risk of AD. This shows a risk of dementia onset in cognitively normal subjects with and without ApoE4 during follow-up of 6 to 9 years in our Nakajima studies. When our risk in E4 non-carriers is set to 1, the risk in E4 carriers is more than 3 in all people. Interestingly, in men, E4 carriers have little increased risk compared with non-carriers, just 1.15. On the other hand, in women, E4 carriers have 5 times more risk than non-carriers. There is interaction between ApoE4 and female gender. May I have the next slide? Yes. We have searched for dietary factors that prevent cognitive decline in people with especially in women with ApoE4 in the Nakajima study. They had normal cognition at baseline. At baseline, we checked dietary habits, nutrients in blood, and ApoE genotype. Can you click the slide? Yes. In this study, we focus on antioxidant vitamins, vitamin C and E at baseline. At baseline, they were. Can you click the slides? Yes. They were 70 years old in average, and after follow-up of, can I click more and more? Yes, yes. After follow-up of about 8 years, they were 80 years old in average. Some of them had MCI or dementia. Okay, next slide, please. Yes, this shows blood concentrations of vitamin C at baseline and risk of future cognitive decline. That is, future onset of dementia or MCI in cognitively normal women with ApoE4. Interestingly, higher blood vitamin C concentrations are associated with significantly lower risk of cognitive decline. This association is not found in men with ApoE4. Our results suggest, for the first time, significant beneficial effects of vitamin C in reducing the risk of cognitive decline in women with ApoE4. That is the highest risk group. May I have the next slide? How had the work been performed? On the basis of our hypothesis that vitamin C can reduce major genetic risk for AD related to ApoE4 and female gender, we have performed 2 studies to elucidate effects of vitamin C. First, a molecular epidemiological study in the population-based cohort, the Nakajima study. And second, experimental study with human ApoE knock-in AD models. ApoE and female gender have interaction and accelerate pathological process of AD. Can you click? Yes. Vitamin C may suppress this process. The point of action may be related to A beta, tau, neurodegeneration, or neuroinflammation. That can be a new therapeutic target of AD. Next slide, please. One more click. Okay. This shows a molecular epidemiological study in the population-based cohort, the Nakajima study. We examined oxidative stress markers. We measured plasma carbohydrate proteins, PCs, as a marker of oxidative stress. The plasma levels of PCs are not correlated with risk of cognitive decline or vitamin C concentrations in women with ApoE4, suggesting that beneficial effect of vitamin C in reducing risk of cognitive decline in women with ApoE4 cannot be simply explained by reduction of oxidative stress measurable by blood. Next, we studied the genes related to vitamin C transport to the brain. Here you see three functional polymorphisms in the 2 genes. Odds ratio of cognitive decline in SET2-1 for ApoE for non-carriers. As you can see, some functional polymorphisms of the genes related to vitamin C transport to the brain are significantly associated with ApoE4 related risk of cognitive decline, suggesting an important role of vitamin C transport to the brain. May I have next slide? This is the last slide. For the experimental study with human ApoE knock-in AD mouse model, we have started to generate human ApoE knock-in AD mouse models. In addition to APP and human ApoE E3 or E4 knock-in, we have to knock out SMP30, an enzyme necessary for endogenous vitamin C synthesis in mice. We humans don't have this enzyme. We first make double transgenic and then triple transgenic model. Using this, we will evaluate effects of vitamin C on learning and memory and neuropathology. May I have click? Yes. Can you click? Yes. We have already generated double transgenics, but generation of the triple transgenic is still ongoing because the COVID-19 pandemic restricted our activities of our animal laboratory, and it's behind schedule. Okay, that's all. Thank you very much for your attention.
Thank you, Masahito.
Yeah.
This is a very interesting topic, and we would like to ask whether there are questions from the floor.
Yes, quite nice talk to know about the vitamin C effect, Dr. Masahito. Yeah, I'm quite surprised, like why vitamin C is not working for the men. Is why is any molecular differences? Vitamin C is good for women, not for men. I can understand there are some biological process for aging, like for some body. Yeah. So, can you comment some point here?
Yeah, that is our question. We don't know why our vitamin C is not effective in men. There are all possibilities. It can be explained by just hormonal, sexual hormones, but it can be explained by any genes related to sex chromosomes. There are all possibility still remains. Yes.
So your mice is like your knocking mice is a male or female?
We are checking both male and female mice, and we will compare.
Okay.
And we will compare the effect of vitamin C between male and female mice. But we still don't have the results.
Thank you, Professor Masahito. Thank you so much.
So thank you, Mohammed, and for your question. And any other questions from the floor?
Oh, yeah, yes. I had one question.
Please.
Are there any other enzymatic activities with SMP30 that could impact the study?
You're asking about other activities, other enzyme activities related to vitamin
SMP30
synthesis. Yes, our SMP30 is our enzyme. Mice have the SMP30 for synthesis of our vitamin C, but we humans don't have SMP30. So we don't have enzymes for synthesis of vitamin C. So we need to take vitamin C from foods. Yeah, we don't have.
Is the mouse SMP30 only for vitamin C synthesis, or are there other functions that can affect your result?
I don't know. I'm not the specialist of vitamin C, but after knocking out by our SMP30, we need to feed mice with vitamin C to survive them. Yes.
Thank you.
Thank you, Masahito.